already recording. Okay, so what I was saying initially is that we are going to do histology, that is the study of tissue. And I've already given you background information what is a tissue. And the great thing is that we also are informed about tissue. So here what you need to obtain are the insight. And the insight about the process of entire thing is that we're going to discuss it together, right? From the first one to the last one. Actually around 5 lessons, a cluster of 30 minutes each. So by around 9 something, we'll have the same exams in our groups, and this one will be an exams between clinical medicine students and nursing students, because the tallying will be done, and after that the entire thing is that you are going to be able to see which class is winning, because we are going to add all the and find the mean average of the entire course. Class of students who are going to participate from nursing, from clinical. Yeah. So you're not seeing my presentation. Somebody saying like that, eh? Okay. Let me give you an insight. I hope right now you can see it, eh? Just a brief one like that. So we want to talk about these things, eh? We want to talk about these things. We don't need an entire notes for us to learn. So what you are seeing there, and I hope Pamela Ogola, can you read for us what you are able to see?
Okay, I'm reading. The study of tissues.
Okay. So thank you. That is confirmation that we are able to see what is displayed. So don't complain that I haven't displayed. That is what we're going to have, right? So
Yeah.
now we have already known that we have four classes or primary classes into which all adult tissues are classified, right? And without any much ado, I want somebody to just tell us or try, you can text if you don't want to speak, eh? Just try and tell us these classes again and again and again. We have four primary classes into which all adult tissues are classified, not infant or a baby, but for adults. Eh? And you are informed about the tissue. Who can state for us the four classes, the main classes of tissue that we are going to discuss today? And I'll be very happy if you do. Or should I mention you? Dan Oxygen, come up. Tell us the four classes of tissues. That's wasted of time. Fahad. Okay, you are not getting me or somebody. Melanie Davido, Neymar you, Nicola Gendo, Diambo Jacqueline, Pamela Gula. Somebody tell us the four main primary classes into which adult tissues are classified.
Can you talk of the connective tissue, skeletal tissues, whatever, those ones?
Those are what I need. Those simple ones are what I'm. That's the main classes of these. Okay. However, epithelial, connective, I can read Neymar. However, the question can be so bombasticated, but the answers are always that. Those ones which you know. Eh? So yeah, after classifying them, we are going to know details about them. Then we are going to talk about the three embryonic germ layers. And what are these three embryonic germ layers? Something to do with the germ. You feel like a primary cells, like sex cells. We call them the gametes, the germ layers. So what are these embryonic germ layers that tissues originates from?
Ni hizo ma mesoderm, ectoderm na endoderm, mama.
Yeah, ni hizo mesoderm, endoderm. Yeah, then gani imebaki? Ama hiyo ama iko sawa.
Ectoderm, sorry, ectoderm.
Okay, sawa. We have the endoderm, the mesoderm, and the ectoderm. And then we have something called mesenchyme, and you'll see about the mesenchyme. Eh? Are you able to see me? My video? Oh,
No.
thank you. I just wanted to confirm that. Okay. A tissue is known to be a group of similar cells, and yeah, they are products arising from the same region of embryo, and then they work together to perform a specific structural or physiological role in an organism or organ. So before they attribute to the entire function of organism, that is you, they have to play a specific role in an organ. And then we have the four primary tissues, which we call epithelial one, connective, nervous, and muscular tissues. We don't have skeletal tissue in the primary classification of tissue, and this is a question that disturbs a lot of students. Right? And then there's this thing called matrix. Who can tell us what is matrix? When I say, well, if I say anything, you'll have known it. What is matrix? Because you will meet many other times you will meet some terms like matrix, then like oh, what is this? So in layman language, Pamela Ogola, can you tell us what you understand by matrix? We are all learned here, and even if you try and then you fail, just a progress at its core. What is matrix? Because there are some drugs, there are some drugs which affect the collagen matrix, connective tissue matrix. So what are these matrices we talk about? Okay, like in biology, when you remember transport in plants and animal, you had the stem cross-sectional view of a stem or a root. You could remember things to do with the parenchyma, sclerenchyma, yeah. So these are the connective tissue, but within the layer of each of them, that meshwork, right? That is what we call matrix. So the extracellular material that compose and surround the tissue, they are the matrix. So when you're told that a typical TB affects the parenchymal matrix of lungs, it has no any strange or miraculous term. It does something to me, the composition of those parts. So the matrix thing, right? Like when you say the peptidoglycans, I think. Yeah, and then we had this one which most of the microorganisms like bacteria, they use it in synthesis of their cell wall. You need to know the mosaic stance and all that. So let's go on. The tissues, they differ from each other in terms of function of their cells, depending with which cells ni cell gani inatengeneza tissue. So when a given cell is the one which is responsible for formation of a cardiac tissue, or maybe, yeah, let's say cardiac tissue, the same cell is supposed to play a different role because cardiac will be performing a different role from kidney. So the cells which are forming tissues in the kidney and the ones in the cardiac, they are different because the cells are completely different. But it doesn't mean that now the mitochondrion is not in this one. No, no. It just things to do with the chemical and the molecular analysis. Now, we are going to see the space occupied by all the matrix. Now, so in example, when I say in muscular and epithelial, the cells are so close, right? And then what is that when you say the cells are so close? It has something to do with the matrix. So when the matrix is so close, the cells become so close. And then when the matrix is scarcely visible, like in connective tissue, the matrix usually occupies much more space than the cells do. And so I wanted to know this. Matrix is composed of something to do with the fibrous proteins, and that is what we see like a clear gel when you go to the radiography and then you do the microscopic analysis, and what we also refer to as the ground substance at most. Let's go to embryonic tissues ambayo nani alituambia, Nicole alituambia example ni endoderm, mesoderm, and mesoderm. So in a nutshell, because I don't want to speak alone, I want one person to take us through the embryonic tissues, what you know, and then I'll give a final ideology behind it. What do you understand by the word embryonic tissue? And what are they and what is their responsibility?
Okay, let me try. I think hizo ni okay, ni vitu zenye tissues, okay, different tissues, like different parts of the body, like okay, parts of the body are made up of tissues and stuff. So like I think ni points zenye hizo tissues zina develop from, for instance like the endoderm. Endoderm ina form hizo internal parts kama like hizo digestive tract and alafu ectoderm ni vitu za nje kama skin, you get like kitu kama hiyo. Sijui explain vizuri.
Yeah, nitakufunza di shortcut ile utaisahau nayo. Unajua tukiwa kwa I don't know, what do you call uterus in Kiswahili? I don't know, is it utumbo or something else? There's a lot of, during embryogenesis, there's a lot of activities which takes in control. Like not, it doesn't mean that besides the fact that we come from two germ cells, the sperms and the ovum, that all the tissues arise from the same process and mitotic division and all that. No, at times one main pigment is formed or a molecule is formed, and then this molecule generate diversified tissue and the other one generate diversified tissue. So like in human development, it begins with a single cell, yes, that is a fertilized egg. And then later on, it soon divides to produce scores of identical what we call the smaller cells. They are very small indeed. And then the first tissues appear when these cells start to now organize themselves into layers. And those are the first two layers, and then sooner or later we have the third there is something which you call strata, and all you know about even in geography what we meant by the word strata, right? That's layers. The first and the third strata, we call them the primary germ layers, and that is what our sister Nicole was trying to explain to us. So all these primary germ layers, they are the ones now giving rise to all of the body's mature tissues. And we have the three germ layers, ambaye Nicole amesema we have ectoderm, mesoderm, and endoderm. I hope there we are good. If you're writing anything, you can write. So the ectoderm is always from the word ecto, near nje. So ectoderm is an outer layer, and then it will give rise to things which you can see from. So kama umechanganyikiwa kwa mtihani and you don't know what comes from the ectoderm, just think it is external. So what do you see in your external your external part of the body? First of all, we have things to do with the epidermis, that is your skin, yeah. And in the epidermis, we have nerve fibers in the skin. So definitely, you say it produces skin, epidermis, and then we have the things to do with the nervous system. And then we go to the second one, that is endoderm. Endoderm, where do we find endoderm? Inside, from the word end. So it gives rise to mucous membrane, which are lining the digestive organs. So don't forget that, right? So we have the mucous membrane from endoderm, and the mucous membrane of the inner lining, the epithelial lining, is the one which now protects from maybe cover the digestive and respiratory tract, and even the glands, right? And then we have the mesoderm, that is the third, which is more loosely organized. So if you want to remember, maybe think about what comes from the mesoderm, think about the most loosened organs, right? So like in mesoderm, the way I've said, they will turn into what we call the gelatinous tissues, which we now prefer. The name is mesenchyme at most, which now will give us some of, but many other organs, like connective tissues, the blood, among like bones, muscles, and etc. Those are coming from the mesoderm. So at your free time, you can go and list them accordingly and keep yourself on toes, because in one way or the other, if you're in first year, you will find them in exams. So most organs are composed of the tissue derived from the two or more primary germ layers. And then the rest of this, we are going to see them how they are going to mature as we progress with the class. Now, one thing is that doesn't mean that because ectoderm will be producing, let's say, skin and nervous tissue, doesn't mean that ectoderm cannot also produce some tissue which are found in the kidney. So the fact that it produces a categorical tissue doesn't mean that the tissue cannot be shared in the organ. I hope we are good there. So the next topic in this one is that we are supposed to interpret tissues, sections, and epithelial tissue. And now here is where we are going to remember what we did in high school, things to do with the temporary and permanent slide. And specifically, if you're going to specialize in things to do with histology, you will be now histologist, and therefore you're supposed to be able to prepare an intact, complete, and potent, slide for view. Maybe when it is for forensic purpose, or maybe for any medical issues, the analysis. So for the histologists, that is, they normally use a variety of techniques for preserving and sectioning, the way you just did it in high school, like even for staining. I just want you to at least text, if you are able, the staining agent that you can still remember. The dyes. No, I don't mean the grams dyes. The staining ones. The ones that are used for slide in the laboratory. Are we good? Can somebody say anything? Guess anything if it is not. Please. Hello? Okay, we don't remember any dye, so let's proceed so that you don't waste time. Okay. So, and somebody, can you please tell us what is called fixation or fixative? What is a fixative and what is fixation? Okay. What do you understand by those two things? Fixative and fixation? Okay. So fixation is a process of fixing a tissue. And what is a fixative? When we preserve the tissue, because at most the tissue are not supposed to be left outside. And I want to ask you one thing. What do you understand by the word autolysis? Autolysis. Somebody can tell us what is autolysis? What do you understand by the word autolysis or autolysis? What do you understand with the word autolysis? Okay, let me mention anybody at random, please. Najma Mohammed, I don't know if you went to Mingia saizi. Faith Tuwai. Faith Tuwai,
Lysozyme.
what do you understand by autolysis?
I think autolysis is when tissues degrade themselves using their own
Yeah, most probably you're right. Very, very right. So the entire thing is that at most our tissue are not supposed to be subject to some conditions which or any other person with a different ideological give us. I mean, if you have an opinion or maybe an explanation which is different from the one which Faith or Tuwei has given us. But that is the third thing. So we have enzymes like lysosome, they will play the role. Tissue are in a state where they can't sustain themselves. They try to degrade themselves, they digest themselves, so that is autolysis. So because when you're preserving this tissue, that is what we refer to as now preservation of the tissue. When we want to use them to observe or maybe for investigation in the laboratory, we have to do what we call fixation. So fixation is just a method where a fixative, and therefore it brings us to what is a fixative. A fixative is what a chemical for example, which one is commonly used in our common, I mean, regional hospital and the morgue? Which one gani inatumika sana? A fixative used even in the morgue. Which one?
Formalin.
Formalin. Thank you, formalin. Nicole, for Christ's sake, it is formalin. Don't forget, so formalin. Formalin is even there in the process of distilling alcohol at most. Formaldehyde. And again, we have other component of formaldehyde. I mean, in some alcoholic reaction, when you take alcohol, and they are incomplete, depending with the diet which you took. There's some probability of formaldehyde being formed. That's why some people die because of alcohol toxicity, not just because the alcohol was bad, but because of the reaction. So that is that. Now, when a fixative is mentioned, just know of a chemical such as formalin that can be used to prevent decay. So then the fixation is the process of now applying that fixative. So then after fixation is done, most tissues are now going to go into what we call the sectioning or what we call histological section. So histological section ni ile tu ya high school ile zinakatwa and remember you need to know that the scalpel must be very sharp to prevent distorting the tissue and most probably why do we normally add water to the component of the tissue? Okay.
for rigidity.
For what? Come up again, Dan.
Rigidity.
Yeah, we can do that for rigidity. Another one is for what? There are two reasons. And there are more than two reasons. Yeah, when you are mounting, you can use water also to make it stick on the slide. Again, you can as well use water for rigidity to also preserve the live state of the cell. So that is that. After that, we take it to the microscope, then we mount it, review it, and so on and so forth. Now, somebody can ask you to define epithelial tissue, connective tissue, nervous tissue, and muscular tissue. Just know what English you can use in that case, right? Yeah, I'm just telling you, sometimes these simple things can come and then, yeah, you start defining epithelial tissue like tissue composed of layers of closely spaced cells. Yeah, you need to have a very great English. Then we do staining. I know you know the main purpose why we do staining is to bring out a finer detail. Yeah. So when you do histological staining, we are just in practical there. We do artificial coloring, like artificial coloring of the slide. So if they were not stained, most tissue will appear very pale, maybe a different color. So when we see, okay, when you may see pink cytoplasm or violet nuclear and blue, green or golden brown protein fibers, depending on the stain that you use. So primarily when you are using a microscope to establish a diagnosis, you stain the slide or the specimen you're using. If it was tissue aspirant, I mean you've aspired a tissue, maybe it's a biopsy, you have a section of a tissue, liver tissue, you stain, and then the primary dye which is going to be used depends. It depends with what is expectation. If you're targeting the nucleolus, this dye is going to bring this kind of color in the nucleus, and that is what we do. So I'm trying to give you all these ones. I want to give you all these ones, and then I need to give you some images at least. Maybe you can recap, maybe see what we are discussing at detail. So why are we talking about all these things after all? Just to remind us of what we're supposed to be having. Now, we still have simple stratified epithelial, but I hope that in this region, all these ones, the things to do simple epithelial, stratified epithelium, you've done them for quite a duration. So I doubt if in epithelial tissue, but just to brush over, when we talk about epithelial tissue, I wanted to be able to describe the property that distinguishes epithelium from other tissue in the very classes. And then you need also to list and classify 8 types of epithelium. There are very many, 8 types of epithelium, and then you distinguish them from each other and state where each type can be found in the body. You also are supposed to explain how the structural difference between epithelium will relate to their functional differences. Because a question can come, explain how the simple epithelium is different from, simple epithelial columna is different from simple epithelial cordial. So you have to know in terms of structural and functional difference. And again, another objective when you're doing epithelial tissues, you need to be able to visually recognize, like in the previous yesterday's discussion, what we were having as a problem is because we were not able to recognize the type of tissue from various specimens or photographs. And that is so bad because I have seen an endoscopy image showing the gastric lumen, especially the epithelial of the gastric. It was brought in the hospital, and this patient was suspected to suffer from gastritis. And there are so many ulcerations in the gastric lumen. And again, the problem comes here. You cannot identify which kind of tissue, which type of tissue. And so your diagnosis is wrong. And once a doctor makes, or maybe a clinical officer or a nurse has an improper diagnosis, the entire impaired. So that is the reasons behind all these ones you are doing. And so when I say you are supposed to be able to visually recognize each epithelial type from special photography, I wanted to know one thing. Normally, most of the epithelial tissue will consist of a flat sheet of closely adhering cells. When I say adhering, they are very packed. And one more cells may be thick, or maybe one cell thick, or maybe more than one cell thick. So mostly when you check on the upper surface, you will see some exposed environment and some internal space in the body. But epithelium covers the body surface and most of the body cavity. So it will cover the body surface and the lining of the body cavity. And on the external ground, it can form the external or internal lining of many organs, even the external lining of the organs. Not only do you need to expect the epithelium to be inside the organ, but even out of the organ. Some organs have the epithelium lining. I need you to know that. Now, I want you to know this. We always have something to do with the epithelium, but between an epithelium and the underlying connective tissue, there's always a layer. And this one normally comes in MCQ exams. So just for this time to ask you, which layer do we get between an epithelium and the underlying connective tissue? What is the name of that layer? Let's say a kidney is always connected to the peritoneum organs by some shaft and fibers. So and then we have an epithelium membrane. And now between the epithelium membrane, the peritoneum cavity, which lines the peritoneum cavity, and the connective tissue, is that membrane. I was almost mentioning it. What is the name?
Is the basement membrane?
Wewe masikio yako. Okay, you're right. It is the basement membrane. Okay. Okay, so okay. I just still get another question. So that is a basement membrane. Don't forget about that, right? Now, mostly the basement membrane is usually too thin to be visible with a light microscope. So when you are using a light microscope to give an investigation, maybe examine a tissue, and then you come up with whatever, maybe you printed the photograph itself, and then you are explaining it to a colleague or maybe a senior personnel, and then you're like, these are best methods that I love to. But it is not suddenly impossible. At most, it can be also possible, right? So that is that. So the basement membrane also contains collagen at most, and that we know that what is a collagen, most connective tissue has it. And we also have adhesive glycoproteins, which we call the laminin and fibronectin. Later on, the large protein carbon complex we have, which is called the heparin sulfate. And all those ones are what is compound of basement membrane. So the basement membrane will serve one main function. But let me ask you, what does it do? What does it do? The basement membrane, mostly. What is the function of the basement membrane? Okay, normally anchor the epithelium to the connective tissue below it. That's that supporting purpose. And then after that, now the surface of the epithelial cell that faces the basement membrane is what now we call the basal surface. So we'll still have basal surface, the one which now covers the basal membrane. And then the one that faces away from the basal basement membrane is what we call what? Who can tell us the part of the epithelium that faces away from the basal membrane? What is the name of that? It is called apical surface. So the one which faces the basal membrane is what we call the basal surface, and then we have apical surface. Yeah, the spelling is just apical surface. So let us now go. Why do we classify and how do we classify epithelial tissues? Somebody can tell us about how do we, in fact, that is the how do we classify the epithelial tissues?
Okay.
Abraham Kiptanui. Nicole Agendo, wacha Abraham atuambie.
According to shape of the cells.
Ongeza volume. Okay, so Abraham, shape, number of layers. Okay, shape. Guled Ibrahim is saying shape. Okay, that's right. Okay, I need somebody who can come up vocally and explain to us. Nicole Agendo.
Okay, according to shape kuna zile zenye zinaka flat shape, hizo ni zile squamous, alafu kuna zenye zinaka cuboidal, zinaka like cube shaped. And kuna zile zinaka longer than they are wider, hizo ni columnar. Then according to layers, kuna zile zenye zina form a single layer, like inaweza kuwa iko either squamous ama cuboidal ama cuboidal ama columnar, but ni layer moja, hiyo tunaita simple. Then if it forms a number of layers, hiyo ni stratified. Then sasa kuna pseudo stratified, ile yenye sasa imechanganyikana.
Unaona vile sasa hizi vitu mnajua kumbe. You guys are so good, eh? So that is that. Now, it is just the way she has put it, but say that you need also to understand, apart from what Nicole again was told us, you just also need to know that epithelial are classified into two broad categories, either simple or stratified. That is the main two broad category, either simple or stratified. So in each category, we have four types. So we have four types in each category. In simple, four types, and then in stratified, four types. So in simple epithelium, most thing that you need to know about simple epithelium that they are the cells, every cell touches the basement membrane. Every cell touches the basement membrane. Whereas in stratified epithelium, some cells rest on top of other cells. Why do we get the word stratified? I mean, it is more than one layer, so you will never find it directly linked to the basement membrane. So before you reach the basement membrane, you'll find a layer piling over another layer. So in simple, they're directly, so they are touching every cell that is the basement membrane. But now when you come to stratified, there are several layers. That is that. Right? Yeah, I hope we are going on well. Very, very well indeed. So let's proceed. Another thing that I need us to understand is that I don't know. Are you able to see the image I've shared to you? There's an image I've tried to share there. I've tried to share an image. Mnaiona? Hmm? Are you guys? Oh, you're not seeing it. Let's try again. Okay, I'll just share the entire image after the class because I don't want to keep on loading what is impossible. Then it will take us time. I will share the image of the simple epithelium, stratified epithelium. Then you see how they look like. So that at least it can be very easy for us. Okay, let us start with simple epithelial and then give it a try of a brush up. Mostly or in general aspect, they have one layer. That one we've already said. However, however, when you talk about pseudo, something to do with pseudo stratified type. What do you mean by what do you understand by the word pseudo stratified type? The one, let me type it here. Maybe the P is always silent, eh? Or somebody can type what I'm saying. I'm tired of typing. The one which called pseudo stratified type or pseudo stratified. What do you understand by that?
Word pseudo means false. Like,
Oh,
so
mhm.
stratified means like it's like confused. Maybe okay, sijui pseudo means false. So like kuna venye tatoi kukuconfuse like imechanganyikana or something like that.
Changanyikiana. Okay, down.
I think I think pseudo strat- I think
Yeah,
I'm not able to get you. Am I the one who is in the problem?
pseudo-stratified, they give a false impression.
Or they give a false impression, yeah? Inadanganya. Eh? Any other person? Could Ibrahim hand?
pseudo stratified is it is a false. It looks like a false number of layers, but it is just one layer.
Oh, okay. So it can deceive that it's a false. Okay, I'm getting that. In fact, the gold Ibrahim, thank you so much. But all of you are right. You're very right. Yeah, that's the point that I wanted. So you see, I wanted to say that generally simple epithelium, they always have only one layer of cells. Although this is somewhat debatable point when we talk about stratified. Because they deceive you as if they are certified, but in real sense, just one layer. Okay, away from that, we have three types of simple epithelium, and they are named for the shapes of their cells. So the simple squamous, and when you say squamous, what do you understand by the word squamous? Somebody please, squamous.
It's flat cells.
Very good. Squamous means flat cells or thin scaly cells. In fact, the one which is so important when I say thin scaly cells. What about cuboidal? Simple cuboidal?
Black cells.
Sijasikia vizuri. Sijui kama internet yangu ni mbaya. Okay.
The cells making the tissue up are cube shaped.
Yeah, they are cube shaped. They are square. Sometimes they are somehow looking round. You know, to find a cell which is square is very hard, as if God came up with a ruler. So when you're going, okay, in the sense of how they name them, let us talk about when it is exams, give square or cube. But in real sense, when you're doing your own analysis with the microscope, don't expect something. Some of them look somehow round, but oblique in nature, they look squarely. Yeah? And then we have simple columnar. What do you understand by what does that mean? Simple columnar?
Rectangle shaped cell.
Yeah, but they're never rectangle. Like, what you need to see when you see is that they look tall and narrow. So they're like tall and narrow cells. Eh? Yeah, Ranjero, you're responding right now. Thank you. Now, good. Let's proceed. There is another one which we call the fourth one. And the fourth one is what we said pseudostratified columnar. It is not under the stratified epithelium. It is under simple epithelium. Yeah. So what I need you to know is that not all cells reach the what we call the free surface. The shorter cells are covered over by the taller ones. So it's like zina hang. So don't expect all of them to be on the surface. So this epithelium looks stratified in most tissue structure, but careful if you use examination, a careful note, especially when you use electron microscope, they will show us if there are every but they are not that way. In pseudo-certified, know that all cells are not reaching the basement, but when you use electron microscope, itakudanganya. So that is why we call it pseudo-certified. So I need you to know that simple columnar and pseudo-certified columnar epithelia produce protective mucus. So when you are told to give an example of where we can find simple columnar and pseudo-certified columnar, just know in the tissues or maybe in the lining where mucus is being produced, because their main, most often reason, zinakwanga secretor, they produce protective mucus coating over the mucus membrane. So this mucus is secreted by wine glass shaped cells. And what are the name of this shaped cell? What are the cells which produce mucus? Zainab Abdul Karim.
Goblet cells.
Yeah, the goblet cells.
Cells.
Yeah, the goblet cells. Thank you guys. Thank you for that. Goblet cells. I know this one is from high school. So I share to you image and then you see you have a cleaner angle of what I mean. So let's go directly to stratified epithelium and then we finish this one. As from your hand, you can see on your presentation, we still have similar one, but ziko ngapi? Moja, mbili, tatu. We still have three page of slides like this one. So after this one, you will have a break if you like, or you can ask questions pertaining the stratified epithelium and so on and so forth, or you can take 10 minutes break, or we can continue also if you're not. So let's talk about stratified epithelium. One thing about stratified epithelia, they range from 2 to 20 or more layers of cells. So they're like stratified layer after layer. So with some cells we are going to be resting directly on others. But only the deepest layer of a cell will be resting on the basement membrane. And I'm going to make a very sweet exams. You just tell me when you want to do it because I feel like this content is so good. So even diagrams will be in the exams. Then you will answer it. That is that. Now good to go. So say is that not all the layers are directly in contact with the basement membrane. So three of the stratified epithelia are named also depending on the shape of the surfaces of their surfaces, like one stratified squamous, stratified cuboidal, stratified columnar epithelium, and that one we have already known why do we give them those names. And then I've said that the deep cells, however, may be a different shape than the surface cells. So don't expect that because it is stratified squamous, the deeper cells will also be squamous in shape. The deeper cells can have a different shape, right? Yeah. And then we have the fourth type of stratified epithelium, which we call transitional epithelium. So I want you to tell me, where did the name transitional epithelium came from? Yes, it go under stratified epithelium, but why is it that doesn't have what we call, the stratified? Or maybe you can add stratified transitional epithelium, but I just need you to tell me where did we get, or maybe where did the owners of the nomenclature behind naming gave the name from? Transitional epithelium. Someone? Yes.
Is it because they resemble both stratified squamous and stratified cuboidal epithelium?
You are very bright. Zainab Abdul Karam, you are not in school right now, you are in holiday, unakula chakula ya mzazi. I could have bought for you tomorrow's lunch. So that is that. Let's proceed. The interesting thing is that it was named when it was thought to represent transitional stage between stratified squamous and stratified columnar. We have, they thought that this transitional epithelium, we cannot just put it in stratified epithelium, but look as if it's something to do with it. It is on a transitional stage between stratified squamous and stratified columnar epithelium. So this is now known to be untrue. The name must be contained. So even though they were challenged, but the name is still contained up to today. Another thing I need to know about what we refer to as the stratified epithelium is that they are very rare, right? My network is gone. I'm sorry. I'm going to put you on right now. Steam imepotea, so Wi-Fi itazima immediately. Hello. Yes, my power